SANDHAN. All Gujarat integrated classroom.
સેટેલાઇટના માધ્યમથી જોડતી કડી એટલે સંધાન.
Hello and welcome to the screen of BISEG. In today's session of BISEG in Microbiology series, we are going to discuss for about an hour the methods useful for detection, enumeration or measurement of microscopic dimension of the viruses. We know viruses very well, that they are smallest kind of microorganisms without any typical cellular structure. And also we know that they are extremely small in dimension, so they are difficult to be visualized by ordinary light microscopic technique. And therefore it needs specialized methods and techniques to study them. Viruses are basically made up of nucleic acid and protein. Some of them, of course, are further encapsulated by lipid material. Now, basic difference between the viruses and other microorganisms is that not only they are smallest in size, but sometimes the largest among the viruses, like vaccinia, are comparable in size to that of the smallest bacteria, like mycoplasma. And they require, unlike prokaryotic counterpart, a living cell within which they grow. So in a way, they are intracellular and obligate parasite. They can exist independently, but as a non-living material. Once they enter inside a cell, they becomes living entity, starts multiplying, being host. Therefore, we cannot cultivate them in a synthetic chemical media like we are doing ordinary bacterial cells. So that is how they are linked between the living and non-living materials. The basic life cycle of a virus is typically beginning with adsorption of a virus particle to appropriate host cell. And once the host cell is recognized, nucleic acid enters into the cell, multiply, and captures the cellular synthetic machinery of a cell, because viruses don't have a machinery of their own. And a viral specific components are synthesized, which are assembled to a mature virion, and a progeny of newly produced viruses are released by a processes like lysis or by way of budding, etc. So let us come to understand that how viruses are studied and how they are important to us, because they cause a lot of disease among human beings, animals, plants, and even microorganisms. So they are important for us to study them, and they are important tools in the hand of researchers as well. In the diagram, we see that the bacteriophage, a typical virus infecting bacterium, and a TMV, a popularly and well-studied plant virus, and HIV, the most studied human virus. So these are typically describing three kind of basic structural differences among virus, although all of them have nucleic acid, either DNA or RNA, which could be circular or linear, which could be single-stranded or double-stranded, but it is always covered and protected by a capsid which is made up of protein only. And you can see in case of HIV that it is further covered by another layer made up of lipid. Now let us understand in the next slide that the viruses are being very much small in dimension, requires special methods to study them. Why we need to study the virus? Because these are important pathogen and they have a close association with human being. Therefore, we need to detect their presence. While studying them, we need to measure their dimension. We need to count their population. So these are the basic requirements. And in today's lecture, we will try to understand how this is carried out. Often we need to diagnose the viral diseases. And whenever we are producing vaccine or some recombinant proteins, then we also need to study them to find out their approximate population and to detect their presence. Although from the point of view of an environmental significance, if we had to assess the infection risk, then also we need to find out their presence. Now, basically, to do so, lots of techniques are available. Well, some of them are time-tested and are traditional techniques which are widely accepted, but they are laborious. Against that, a newly developed method have came up in the recent past, which are modern, much fast, rapid, and are very much precise too. Now, we will try to understand the direct methods in the form of electron microscopy, X-ray diffraction technique, and filtration like physical techniques, which will be aimed at finding out the presence of viruses. And the another class of method called indirect method are based upon assessment of infection causing capability of the virus or finding out their infectivity. And these methods are based upon finding out the presence or absence of antigen or antibodies against viruses, or maybe specific nucleic acid or genomic constituent of the virus. The most popular methods among the direct kind of technologies are electron microscopy. And electron microscopy is, you can say, is a gold standard for the purpose of counting particles. And in these techniques, samples are fixed over an ultra thin membrane and using the chemicals like aldehyde. And afterward, the viral preparation can be stained positively or negatively using compounds like osmium tetroxide or similar salts. In case of scanning electron microscopy, shadow casting using heavy metals like palladium, gold, etc. is used. Now, in electron microscopy, as you see in the diagram, that the beam of electron passes through a very thin sample and an image is formed over a fluorescent screen. And from this fluorescent screen, image could be recorded in the digital format also. The modern softwares used for image analysis purpose can readily distinguish between dimension of different size appearing in the microscopic field directly. But it is really difficult to find out the height of the microscopic object from the image analysis. Now, this can be done by a very simple calculation as shown in this diagram, that a ratio in a shadow cast preparation, a ratio between the object and the shadow in terms of their height and length are always equal. So that is how the height of the object can be found out by a simple formula, that h equals to d into sine theta, where theta is the casting angle through which the shadow has been cast upon the object. Now, in the next slide, we will see that another technique to enumerate virus is make use of fluorescence technique. The sample, when is large in quantity, like environmental sample, drinking water sample, etc., where we have to find out the presence of virus, which are potential threat to the population health, then these samples are analyzed by concentrating the viral material in a very small volume, and the samples are stained by fluorescent dye like SYBR Green, produced by Invitrogen, marketed as molecular probes. So these fluorescence products are in fact antibodies specifically directed against the viral antigen, and these antibodies are tagged with a fluorescent label. So wherever there are viruses, the antibody will bind to viral antigen, and along with these antibody, fluorescent label is also getting tagged to the viral particle. And that is how wherever the fluorescent label has been retained in the sample, we see a bright green colored spots, as we see in the microscopic image over the slide. Now, on the basis of these appearance, we can not only detect the presence of virus, we can also enumerate them, we can know about their morphological features, and by using specific antibody, we can identify the kind of virus also. Now, another way to find out the viral population is making use of proportional count where the sample are mixed with the known number of latex particles, and these latex particle, especially ratio of the latex is to viral particle, is compared, and that is how we can exactly enumerate the total number of viruses in a sample. Now, atomic force microscopy is another technique whereby we can check the morphological features of a very ultra microscopic dimension. Now, this technique is, you can say, very much similar to reading the braille markings with the help of fingertips. In the same manner, a very microscopic dimension probe called cantilever is having a very sharp pin, and that is kept pointed over the object. And this cantilever is moving along the object, as we see in the diagram, that a pointer of the cantilever, once it traverses across the sample, it will move itself, and the migration or shifting within this cantilever is easily recorded by a laser beam which is deflecting its signal, and the collection of deflected laser data is utilized to generate the images. This is a most powerful technique for studying the topographical aspects, hardness of the specimen, and precise three-dimensional organization and structure of an object in the magnitude of a fraction of a nanometer. So this is one of the most powerful technique for studying the viruses. The another technique is X-ray diffraction analysis. We know that the viruses were crystallized for the first time by Martin Stanley in 1935 as a proteinic product, and this was in fact a crystalline isolate. So crystals are best studied by X-ray diffraction like techniques because they have a regularly arranged lattice structure. So once an X-ray is allowed to incident upon any crystalline lattice, the X-ray are deflected as and when it come across the molecular structure in between the path. And this deflection pattern is collected, and based upon finding out the deflection angle called Bragg angle or theta, we can generate the useful information like distance between crystal lattice structures. So this technique is so powerful that up to a resolution of 10 raised to minus 10 meter, that is comparable to that of a bond length, the structural details can be visualized, predicted, and analyzed. Another technique for the study of virus is much simpler, that is technique like filtration, which can be done in any laboratory. The viral sample is allowed to is first distributed over a bacteria-proof filter, and once the sample is loaded, the filter membrane is placed over a culture medium which is previously charged with the bacterial inoculum. Now, if the viruses are able to traverse through the filter membrane, they will be able to infect the host cell over the medium. If not, then will be retained by the viral proof filter membrane. And if a poke-like or plaque-like structures are seen in the cell culture, then we can say that the filter is large to retain the virus, or porosity is too large for the virus to be retained. And by use of successive filters of different and varying porosity size, we can find out that what dimension viral structure is having. So this is simplest, although a little bit time consuming method, but easy to perform in any laboratory. Now these were all the direct kind of methods which gives us an idea about what is the physical nature, or what is the microscopic size, or how to count the particles. But it doesn't distinguish between the particles which are able to cause infection and those particles which are unable to do so. In another word, the method do not distinguish between living or dead viruses. For that, we need to have a specialized technique which are related to infectivity measurement, or measurement of capability of the cell of the virus to cause successful infection. So these different class of methods are largely based upon molecular biology techniques and immunological techniques. We have with us Dr. C.G. Chandran, who is having a specialization in molecular biology earned from Indian Spice Research Institute. So I would hand it over the further discussions to her to describe the indirect methods for the enumeration of viruses. Dr. C.G. ma'am, please.
Good afternoon. Let me start with the indirect methods. Indirect methods includes methods those rely on infectivity, and the methods those rely on serum based analysis, that is antigen antibody reaction based on antigen antibody reaction. And the other method, the most modern method, it depends on viral nucleic acids, the detection of viral nucleic acids like DNA, RNA, and proteins also we can detect using this method. So let me start with the indirect method. First, indirect method, and the most common and the standard method for the indirect detection of viral titer. What do you mean by titer? That is the concentration of virus in per ml of the sample. And this is first developed in 1950s for the titration or to find out the titer of bacteriophages. And later in 1952, Renato Dulbecco modified this procedure for other viruses that infect different hosts, including humans. And in this method, the viral sample can be serially diluted, and from the dilution, 0.1 ml of the aliquots can be used to infect monolayers of host cells, which is already kept inside the Petri plate. And after incubation, and during this incubation period, the virus attaches to the cells, and after